Good afternoon, everyone. My name is Yasmin Rahimi. I'm a senior research analyst here at Roth Capital Partners in the biotech sector. Currently cover 19 names. Half of them are in the liver therapeutics. It's really a pleasure with us to have Jay Luly, President, CEO, and Director of Enanta Pharmaceuticals. Jay, thank you very much for being here. So the format of this session is fireside chat. Feel free to interrupt during the sessions to ask questions. So, Jay, can you briefly give us an overview of your lead assets and their respective programs, and as well as clinical developments and binary events for this year in NASH, PBC, RSV, and Hep B?
Sure. So thank you, Yasmin, for the invitation. But before I begin, I want to remind you I'll be making some forward-looking statements, and for a summary of the risks associated with these statements, please see our filings on sec.gov and on our website. So Enanta is a liver disease and virology-focused company. We have really four main therapeutic areas that we've chosen within that area. So Hep C is what we're probably best known for. We have now progressed two drugs with AbbVie to the market. We are the protease inhibitor in AbbVie's regimens that they sell. So the first one was called Viekira, and now the second one is called Mavyret. And those are commercial stage. We used to work on lots of Hep C programs before that, but that's more in our past. What we've done is now that Hep C has great cures, such as Mavyret, we've moved on to other important areas in liver disease and virology. So we're in phase 2 in NASH, non-alcoholic steatohepatitis. We're in phase 2 in PBC, which is primary biliary cholangitis and other cholestatic liver disease. We're in phase 2 in RSV. Respiratory syncytial virus is another important area for which there are no therapies approved for marketing today, and something that we're very excited about. And then last but not least, going back to the liver, we're preclinical in hepatitis B, but we will be, we expect to be at the clinical stage in the second half of this year. So stay tuned on that front. We have a very interesting core inhibitor program for hepatitis B. In terms of catalysts, I think we just announced today, actually, that we've completed enrollment on 2 phase 2 trials. So those are high on the list of catalysts for the year. The first one is in RSV. That's a short-term treatment study, so it's not so many days after the completion of enrollment that you're done with the actual study. We expect to have that data mid-year this year. And then the second phase 2 study that we completed enrollment in is the so-called Argon-1 study for NASH. That's a 12-week study, so we've got 12 more weeks for the last patient in to get to that, and so therefore the readout for that study is expected at the end of Q3 of this year. And then, as I mentioned, we expect to be in the clinic with our HBV molecule in the second half also, and we expect to announce other NASH candidates later this year as well.
Thank you, Jay. I'm going to go off script for a sec, but for our audience who have not had a chance to meet Jay and Enanta Pharmaceuticals, Jay has done an incredible job of doing something that I have not seen any biotech company do so far, which is walk me through from when you became public, the transition of not only developing the first best-in-class Hep C, but 2 in a row, Viekira and Mavyret, and then followed by sort of the capital structure that has been in place till now that has funded all these additional programs.
Sure. So we went, I've been CEO since 2003. For those of you who were private company investors in the early 2000s, it was a very difficult time for capital raises. So we had to be very efficient with capital. We did it largely through structuring deals that were highly non-dilutive, and that allowed us to survive basically a dark decade until IPOs were even feasible again. So we were considering an IPO in the late 2012 timeframe. We ended up going out in early 13, and we were one of the, I guess, the first IPOs that priced in the range and then traded up and so forth. I think from the capital, what you're referring to is the last equity raise other than the IPO that we did was in 2006. So, we have funded the company in a very highly non-dilutive way. That's allowed us to, get to the public markets, which we really haven't accessed since our IPO. We don't expect the need to do that for the foreseeable future. We've been profitable since 2010 consecutively, and had over $200 million in royalty revenue last year, to fund our wholly owned pipeline. So it took a while to, sort of build up that, not only the R&D foundation, but the financial foundation of the company. But now we're pretty much, at least for now, self-funded and progressing, again, the wholly owned pipeline ourselves.
Thank you. So I think this is quite a remarkable accomplishment.
Thank you.
And especially for going after very competitive indications and being a leader. So let's spend a little bit of time in talking about your NASH program. As you revealed this morning, enrollment is complete. The phase 2A is going to read out end of third quarter. So for our audience, I know in the last session, maybe briefly walk us through how your FXR agonist differs from Intercept's OCA, and how you're planning to move it forward and what sort of efficacy and safety you want to see.
Sure. So for those of you less familiar, FXR is a target that's been worked on in NASH now for several years. Actually, Intercept was a trailblazer not only in this mechanism but in the NASH field in general. So I think they deserve a lot of credit for really opening people's eyes to what the possible could be in NASH. So we're working on FXRs, are they, is Novartis and Gilead. It's not, anytime you have a good mechanism or what we think is a good mechanism, it's not going to be without competition, or maybe not without multiple products on the market, over time. That's our assumption. That's our base case. I think the way we're thinking about it is we just want to be one of the FXRs that's out there, one that's, competitive, and probably most importantly, one that's your ticket into combination therapy, onto which you can add other therapies. So those are our kind of high-level goals, and stepping back from that, we started our FXR program internally. Again, we didn't license anything in. We sort of built it all from the ground up. So we had, on the one hand, no asset to inherit, but on the other hand, we had a nice white sheet of paper that we could decide which chemical classes we wanted to work on, how we wanted to design the molecules, et cetera. So early on, we took a non-bile acid tact, which pretty much is what everybody's done, but Intercept is taking a bile acid approach. We're taking non-bile acid approach, and even within that, you can drill down, and there's multiple different kinds of chemical classes you could get into in the non-bile acid realm. But overarchingly, what we wanted to do is create very potent, highly selective agents that would hopefully be good on-target efficacy and, minimal off-target liabilities from a safety perspective. And from that perspective, we worked diligently preclinically, both in vitro and in vivo, benchmarked along Ocaliva at every step along the way, and uniformly, at least preclinically, we were either more potent and or more efficacious than Ocaliva in a variety of models of NASH and PBC and models of fibrosis. So that's enough to win you a ticket into clinical trials, and we progressed into phase 1 after that. Phase 1 data set looked, very strong. I would say our preclinical goals, at least from a potency perspective, played out clinically. We're roughly at about 1/10 of the dose, I guess, of intercepts is what we've chosen for phase 2s. And now, after having looked at a healthy population in phase 1 and a presumed NAFLD population in a separate phase 1 that we did, we're now into the Argonne-1 study, which is a longer duration study. It's a 12-week study, and one in which we are using ALT as our primary endpoint, and then several other secondary endpoints that will be reading out, things like the usual biomarkers of FGF19 and C4. We'll be looking at MRI PDFF so that we can look at the level of steatosis or fat in the liver, and others. So those will again read out in toward the end of Q3. And then the aggregate, what we're looking for is good directional. It's like any phase 2A that you do. You're looking for directionality on the efficacy markers. You're looking for, that the PK, PD generally mapped with what you knew before, and that you've got a clean, safe dose to progress into phase 2B. So those are the things to be looking at. And in parallel, working on follow-on FXRs, thinking about it sort of generationally, much as we did in Hep C. As soon as we got paritaprevir, the first protease inhibitor, we started barreling down on what became glecaprevir, which was an even better one. So.
Yeah, I think for our audience who may not know Jay and Ananta, one of the things that's probably most intriguing is that they develop everything in-house and not only have one compound, but an entire generation of multiple scaffolds. So like you said, with the FXRs, in-house developed and continued. Gilead acquired theirs, Novartis acquired theirs, Intercept built it in-house. So there's a library, and then you briefly mentioned, but you also are sitting
and then to build it in-house.
You build it in-house, yeah. That's exactly right. You also have built a large number of other undisclosed targets in NASH, of which one recently was disclosed last week, which was Solansertib, ASK1, I apologize, ASK1 inhibitor, which is the same mechanism of action of Solansertib. So can you, to the extent that you can give us, elaborate on how does your ASK1 inhibitor that we're going to see the data at EASL differ from Gilead Solansertib? You can say no comment.
Yeah, well, that we had an ASK1 program was previously undisclosed until it sort of came out in an earnings question. So, but it would have come out at EASL anyway. So we have two presentations at EASL in a few weeks. They're on two different ASK1 molecules. We haven't yet nominated either as a finalist candidate, so let's be clear about that. And we're intrigued by the mechanism. We've done a lot of work. You can imagine we've already messed around with combinations and done other kinds of things. But in addition to ASK1, we're working on other things in NASH as well. Again, when we go into a therapeutic area, we go deep, multiple different targets, multiple different molecules against each of these mechanisms, and that's what we're going to build up over time. So stay tuned for EASL. I think a lot on ASK1 really boils down to what do Gilead's phase 3 data sets look like, not only the one that they've reported out, which was not a success, but the one that they're looking at in F3 patients, so-called STELLAR-3, which may or may not have success. So we'll be looking at that with interest, and if they're successful, it's obviously good for us. If they're not successful, there may be reasons why ASK1 is still interesting. We just need to crawl inside that data.
And then, I mean, you have developed in-house and co-commercialized with, have your partner AbbVie for Bicara and Mavyret. So you've learned over the years enormous amount of combinations in regards to HCV, and now you're going into NASH, you're going into Hep B. What are the key lessons that you learned from the Hep see that it's applicable to other big liver indications?
Yeah, so it's nice that RSV, we may not need to do combination therapy because everything else we're doing is combo. So with Hep C, as you alluded to, we played around with a lot of different combinations in-house, and then ultimately we did with AbbVie too, and we came up with the winning combinations, and now just the 2-drug combo. No one thought that you could do it without a nuke. No one thought you could do it with just 2 drugs, and now it's the #1 selling Hep C drug in the world, even when you compare it to all the other ones added up. So it's successful when you get the combinations right. So I don't know how many of the exact lessons that carries over into other combinations like Hep B or like in NASH, but we feel like we have learned some general lessons in terms of profiling mechanisms individually and then looking for traits in these molecules specifically and mechanisms that lead them to be more combinable, some more combinable than others. And so we do a lot of that work preclinically, but ultimately we do need to do it clinically in the end.
Then maybe we can spend and transition now on RSV. So your developed 938 is currently being developed in a phase 2A RSV challenge study with results expected mid-2019. Briefly walk us through 938's mechanism of action, how it differs from other modalities that's being developed for RSV. And then part B is what efficacy, what do you want to see out of the RSV challenge study again that allows you to give you the go decision to go into a phase 2B larger study? And then part 3, which patient population?
Sure. So a lot of, there is a drug approved for RSV. It's prophylactic though, so it's called Synagis. It's given to babies who are born premature, who have a high risk for RSV infection. They're given a prophylactic monoclonal antibody that targets the F protein, the fusion protein in the virus. After that drug came to market and sold okay, other people started targeting F protein for a potential therapeutic mode, which is once you have the viral infection, can you come in and treat it with an F protein inhibitor of some sort. So monoclonal antibodies along those lines, therapeutic mode, have failed. Regeneron had one, failed. The nanobody is going after, that was fought by Sanofi, is maybe not so encouraging so far, going after the F protein. Some small molecules, Gilead has a fusion inhibitor that went after the F protein. It hasn't looked so good. But there are other fusion inhibitors that are coming forward. Maybe someday one of them will. We're taking a very different approach. Rather than trying to block the virus at the stage before it gets infected into the cell, we worry that if the infection progresses too far, the virus is already inside the cell, and now you've got to deal with the virus that has now set up a replication factory inside those cells. Maybe it's better to come up with a mechanism that targets the virus inside the cell where that replication is occurring. So we've got a direct-acting antiviral approach. We're working on a protein called the N protein. And so far what we found is that it's very effective at blocking replication inside the cells, and it has an extremely high barrier to resistance, which means a couple of good things. Number one, it's never good to have resistance, and number two, it may mean that you could avoid the need for having to do a combination therapy for resistance prevention, which is probably not the case with fusion as a target. Fusion has a low barrier to resistance, and so it's easy to see those kinds of mutations pop. So we're in a phase 2 study. Phase 1 looked great. We had a very clean safety profile. The good news is in phase 2 and phase 3 and probably beyond, you're only going to have to dose for about 5 days. So we've dosed longer in our phase 1 study and established safety in that study than we need to dose in our phase 2 study that's even ongoing, even though, so the enrollment, as I mentioned, is completed, that we announced that today. So we'll be looking forward to that data set. In phase 2, we took healthy volunteers, infected them with the virus. You wait till the viral loads build up to a very healthy state, the people become symptomatic in terms of respiratory symptoms, and then you treat them with one of either 2 dose regimens of our drug or placebo, and you look for differences. What we would expect to see is viral loads to drop, signs and symptoms to improve versus placebo, and these are the readouts, again, that we should have by mid this year.
And then based on that result, which patient population would you be focused on in your phase 2B? Is this the elderly, the immunocompromised, the children?
Yeah, so you rattled off the 3 major buckets. Kids, every kid by the time they're age 2 gets one of these infections, so it's a huge potential market in children. But the elderly are also susceptible, and so are the immune compromised. Our intent is to be in all of those groups of folks eventually, but we're going to start in adults first, learn how the drug ticks, look a little bit more about viral kinetics, learn about just the overall behavior in a patient population in adults before we move into to peds, and we'll have more discrete clarity on that exact patient population as we roll into data mid-year.
Thank you, Jay. I'm going to open it up for our audience for questions. If not, I will have 2 more for you. Jay, I guess the question for you, we didn't talk a little bit about, we should spend some time on Hep B. You've selected 514 that's going to get into the clinic this year. How does 514 differ from its previous prototype? Its mechanism of action in relation to other molecules that are in development.
Sure. So Hep B is a big problem. Hep B has been a problem for a long time, just like Hep C. Unfortunately for Hep B patients, the rate of progress for Hep B has stalled for a long, long time. In fact, Hep B is sort of where Hep C was 20 years ago. The cure rates are very low, maybe 10% with interferon, maybe a couple percent with nukes. You can suppress the virus with a nuke, but you can't, really get to a functional cure in that patient in any meaningful way. And there's been a dearth of other mechanisms onto which to add a nuke. So now that Hep C is, for the most part, a cure story, it's just a matter of time and money. A lot of the energy has pushed over with new vigor to Hep B and trying to figure out, what we can do. So lots of competition over there too, so that's no different than probably everything else we work on. But again, we sort of started with a blank piece of paper on Hep B and picked our targets in terms of what we wanted. And one of the things that intrigued us several years ago was this whole capsid assembly mechanism, the so-called core inhibitors, basically block that viral assembly. And there was a little company called Novira in the early days starting to play around with that, and as were we. Novira showed some clinical proof of concept, although with perhaps not the best molecule, which is always a great eye-opening event if you're in this business, because there's enough there to give you hope, but not enough there to get you probably across the line. So J&J acquired Novira, and then they started building their core program even bigger, so they have some molecules going forward. Roche is in the game, Assembly, Arbutus, us. And so, again, like I said before, if there's a good mechanism out there, going to be multiple people working on it, great. Again, our goal is just to end up with a very solid member, likely not to be the only one, but one that we can use as an anchor to build onto other pieces for combination therapy. So again, it becomes a ticket to the show in HBV combinations. So we didn't progress some of our earlier molecules. We had some very good ones early on, but we wanted to wait until, because already people are talking about, second generation ones and maybe third generation ones coming even before we've really got data on the first generation one. So whatever generation it's being called out there, we wanted to sort of come in at that altitude with our first one. And we were patient. We had a prototype one early last year that checked, pretty much all the boxes. But we had some other ones that looked intriguing, that they might be even better, that we could progress. And we wanted to give ourselves to the end of last year to make a final call. We did. We picked a new molecule called EDP-514. Again, we'll have data on it at EASL in a few weeks, but it's a very strong, solid entrant as our first molecule going into HBV. I don't suspect it'll be our last. You can imagine we're thinking about follow-ons. We're already working on other mechanisms in Hep B that we haven't really talked about yet, and we're thinking about it just like we did with Hep C. The longer-term game, there's lots of chances to build value along the way, but ultimately, the winners are going to be the people who can assemble the best set of assets, build the best combinations, and bring them forward to patients. So,
perfect. Thank you, Jay. Maybe take the time to thank Jay for being here and for a great discussion.
Thank you very much.